Nimistrapalos, nanotoka Karina, nimo machtia yoltecuacuame. This is hello, my name is Karina and I study tarantulas in Nahuatl. I am an indigenous scholar and part of the first cohort of the NSF-funded Bridges to the Doctorate Louis Stokes Alliance for Minority Participation Fellowship Program at the University of Idaho. Today I'll be talking about my research on the Madrean Sky Island tarantulas. But first, I would like to acknowledge that the University of Idaho is located on land belonging to Nez Perce and Coeur d'Alene's peoples, and that our current Sky Island research is taking place on the lands of the Apache and Tohono O'odham people. So some background on the Sky Islands. A Sky Island, as you may or may not know, is a mountain that is separated from other mountains by distance and dramatically different environments between the higher and lower elevations. These mountain ranges have several stacked biotic communities that range from oak woodlands at higher elevation to desert at their lowest, creating forest islands and seas of desert. The Madrean Sky Islands are located in the Cordillera Gap between the Rocky Mountains, Colorado Plateau, and the Sierra Madre Occidental of Mexico. This region contains the most Sky Islands of any other region on Earth. It is considered a biodiversity hotspot given its location between the Chihuahuan Desert, the Sonoran Desert, the Rocky Mountains, and the Colorado Plateau. In this region, a Sky Island is defined as a mountain range that is high enough to include Madrean oak woodland habitat, isolated by dry land. There are 65 isolated mountain ranges spanning the Cordillera Gap. 32 of them are in the United States and make up 21 distinct Sky Island complexes, and at least 33 Sky Islands of which are located across the border in Mexico. The desert grasslands in the valleys have limited genetic interchange between populations of organisms and created the perfect conditions to diversify, not just between the individual mountain ranges, but also within them in these stacked biotic communities. On the slide to your right top is actually Madera Canyon. That is what you typically see as a Madrean oak habitat. And beneath that is the Chiricahua Mountains, seen from down in the Chihuahuan Desert. So onto the spiders. So Aphonopelma is the tarantula of the United States and includes 59 described species across the US and Mexico. In general, they live in burrows with openings beneath rocks or out in the open. They range in color pattern but are mostly black with some brown-red hues. They are found through a variety of habitats, including the Madrean Sky Islands, where my group of interest, the marxi species group, resides. The marxi species group includes 7 described species: A. marxi, A. piloncillo, A. catalina, A. chiricahua, A. madera, A. voorheesii, and the newly described Aphonopelma bucadewachi. These tarantulas likely moved into the surrounding habitats and up into the mountains where they speciated, trying to adapt to the different environments. Tarantulas from high up are not going to be likely to cross the deserts, and so this combination of genetic and geographic isolation caused by the different biotic communities and the differing climate create these selective differences that are likely to promote the diversity of this group. Species delimitation in Aphonopelma overall has been difficult due to the morphological variation in the traditionally used morphological characters and the use of a couple specimens, sometimes from the same area, to describe a species. But in recent years, species boundaries of the United States Aphonopelma have become much better understood, not just through morphological characters, but also using genomics, locality data, and mating patterns. Unfortunately, for a while, samples from the Sky Island ranges were lacking both in historical and modern collections due to the difficulties in reaching their habitat and the elusive nature of these spiders and their burrows. Which is, finding these spiders is really hard unless you know what you're looking for, and even then it's still difficult. Since then, increased sampling of the U.S. Madrean Sky Islands in particular have revealed a number of potential new species, including some at higher elevations than previously thought to be inhabited by Aphonopelma. Sampling in Mexico yielded a new species, as expected, described by Dr. Brent Hendrickson, Aphonopelma bucadawachi. So with only a portion of the Madrean Sky Islands explored for tarantulas, including the ranges across the border in Mexico, and the recent discoveries of Aphonopelma at higher elevation, there exists significant potential for additional undescribed species within some of the higher mountain ranges. So, why study these tarantulas? Why does it matter? Well, in the big picture, it's to help determine the effects of climate change on sky island habitats. How to manage for the effects of climate change on plants and animal species has become of increasing concern for resource management. These tarantulas and other sky island organisms can offer insight into whether species will be able to adapt or go extinct in response to future climate change. There's also potential for incredible diversity in this group. The diversity within the United States makes it likely that there are new species on the Mexican side of the Madrean Sky Islands, where there has been less sampling and more mountain ranges. There's also the status of these tarantulas as a conservation concern. There is not enough information on the population sizes, given that they are really hard to find compared to their lower elevation counterparts. And it begs the question, are they just hard to find, or are there just not that many? But the goal of my research is to understand the evolution of the Sky Islands and how these tarantulas moved into the mountains and got isolated. Answering questions like, was their move into the mountains a single event? Or did the species split up as the Sky Islands formed? Or were there multiple invasions? So on to my preliminary work. We got some sample sequence of 31 individuals of different species in the Madrean Sky Islands. Samples are from 9 different mountain ranges, that being Catalina, Chiricahua, Madera, Peloncillo, Dragoon, Galliuro, Hornbrook, Whetstone, and Huachuca. We used 1,200 loci and analyzed using the PHYLUCE pipeline. Assembly was done using spades. I searched for UC loci using a blended version of the arachnid spider probe set from Kulkarni et al. 2020 and Starrett et al. 2017. And the alignment was done using MAFT and then trimmed using G-blocks. The concatenated alignment was then used for estimating a maximum likelihood tree using IQ tree and a species tree using SVD quartets. So, the relationships between these species are consistent across both methods with good support values. Starting with our specimens from the mountain ranges not sampled before, we have Dragoon and Galliuro samples being sister to each other. We have a Aphonopelma Madera and samples from Whetstone as sister, and Aphonopelma marxi and samples from Hornbrook Mountain as sisters, and having Voorheesi as a sister species to marxi and all other Sky Island Aphonopelma. Aphonopelma marxi was considered to be sister to all these Sky Island species, but as more specimens are added, we see Voorheesi falling out as sister to marxi and other Sky Island species. And here we have Aphonopelma Catalina and Aphonopelma Chiricahua as sister to each other, which confirms results from previous trees in the Aphonopelma revision of 2016 by Chris Hamilton, and after the inclusion of Aphonopelma bucadawachi in Brent's 2019 paper. Something interesting that's going on in this tree is the position of Aphonopelma Chiricahua and a novel species that is in the works, not described yet. Both of these specimens are from the Chiricahua Mountains. They overall look very similar with the exception of their size. And while there are differences in their elevation, there are overlaps between their distributions. This result suggests that if a mountain is high enough, we are likely to find more more than one species at the top of these biotic communities. And it tells us a little bit more about how these spiders moved into these mountains. So for future work, we're going to continue sampling the United States Madrean Sky Islands, especially at the higher elevations. We're going to increase our sampling of the mountain ranges in Mexico, which are not very many, and we're going to work on some niche modeling and some biogeographical hypothesis testing. With that, I would like to thank the Arthropod Molecular Systematics Laboratory, my PI Chris Hamilton, and the rest of the Hamilton Lab, Eric and Andrea. I would also like to thank the NSF LSAMP Bridge to the Doctorate Program at University of Idaho for funding me, and the American Arachnological Society and the Vincent Roth Fund for Systematics Research for funding my future expeditions. And finally, I'd like to thank Dr. Brent Hendrickson and Dr. Jason Bond for sample collection, and Wyatt Mendez for serving as an excellent spider hunter and a great guide. Thank you very much. Hope you enjoyed